Okay, hello everyone. We're still about a minute away from 5 o'clock, so we're just gonna sit there, look at each other, and you get to look at us. While that minute passes, there we are, it's 5 o'clock. Welcome everyone. It's first of all, Happy New Year with, the happy, let's hope that 2021 brings better news than 2020. I hope all of you have survived the holiday period with minimum damage to your health, to your economy, and to your psyche. Welcome back to Worldwide Neurodevelopment. It's a real pleasure and probably rather appropriate to start the new series in this new year with Oliver Hobert, who many of you know needs no introduction. Nonetheless, I think I'm going to introduce him anyway. So Oliver did his PhD with Axel Ulrich at the Max Planck Institute for Biochemistry in Martinsried near Munich, Germany. And then a postdoc with Gary Rafkin. And in 1999, he joined Columbia University. I assume as an assistant professor, and eventually became a professor. And since 2005, so 6 years later, he became Howard Hughes Medical Institute investigator, Howard Hughes investigator. And he's still at Columbia since then. He did move physically, however, he just told me, from the medical school to the arts and science, to the College of Arts and Science in 2016, where he has, and this is something few of us do, voluntarily added to his teaching load. He's telling me about a very cool course that he teaches in neurogenetics. So Oliver has really made seminal contributions to a key topic in developmental neuroscience, which is how neurons adopt and maintain their terminal identity, right? And he coined I believe you coined the term terminal selector genes, which are genes that are necessary and sufficient to endow a certain neuron with a terminal, what is called a terminal identity, often involving the type of neurotransmitter it releases, but can also involve its shape, its connectivity, and so on. So it's obviously not trivial that a neuron should distinguish itself from other neurons. Many of us try to understand the basis of this distinction. It's even less trivial how you maintain that in the face of a constantly changing and challenging environment. And Oliver has studied in particular the transcriptional code for how this works. He has revealed not only the combinations of transcription factors required for this, their hierarchy, but also the regulatory genomic landscape that contributes to this, that is read by those factors. And when there are exceptions to these rules and what the rules are and so on and so forth, or what the rules are and when there are exceptions for them, how the exceptions can be explained. And I should say that the reason this is seminal really is because it also triggered a whole field, essentially, of trying to understand to what extent are the rules the same and the nuts and bolts of those rules similar or different between the model organism that Oliver has established this paradigm in, which is C. elegans, and so-called more complex, but really just numerically larger and models in Drosophila and to some extent in mouse as well. And I think it's nice to see that essentially the rules are not really that different, but the way they get applied and the nuts and bolts and the nitty-gritties of them tend to vary depending on the extent of what you might call non-hardwired events that contribute to the development of these organisms. So it's really not just uncovering an important piece of the developmental puzzle, but really laying the grounds for the expansion of this work. And for this, I think Oliver deserves a lot of credit and a lot of kudos. So without further ado, Oliver, it's a pleasure to host you. The floor is yours, and whenever you're ready.
Great. Thank you so much, Bassam, for the great introduction. Very nice introduction. Let me share my screen and let's see. Bassam, does it look good?
Perfect.
Okay.
Ready to go.
I heard comments. I saw one comment about noise issues. We also had this in the beginning, Bassem and I, when we started, and we just logged in and out and refreshed our screen, and then it eventually worked. So, but nevertheless, do speak up. I have to say though, I can't see anything but my slides, so I would need to ask Bassem to interrupt me if there's any issue. All right. So so again, thanks for having me here. It's great to have sort of such a broad reach with this type of platform, and this is, I think, one of the very few positive things that we'll take from this COVID crisis, that it somewhat democratizes the scientific community and allows many more voices to be heard, but also to broadcast one's voice much more broadly. So what I want to talk today about is I want to talk about the studies that we have engaged in over the last many years in trying to understand how terminal differentiation programs in the nervous system are genetically specified, as Bassem already mentioned. But I want to sort of highlight one specific angle here that has always sort of fascinated me and motivated me in regard to studying this problem in C. elegans, which is that because of its limited complexity, 302 neurons, 118 different types, or anatomically distinct classes, I should say, because of that limited size, one can really envision and hope to look at this problem of terminal differentiation, not just on the level of some restricted parts of the nervous system, but that one can really look on a systems-wide level and hope, and one can hope maybe to carve out common principles that don't just apply to some parts of the nervous system, but are used throughout the entire nervous system. And this sort of global systems-wide perspective is something that we have really been always striving for. And I want to tell you today about some of these principles that are emerging. And I'll give you a disclaimer right away that much of what I will be talking about, particularly in the second part of my talk, is involving a lot of speculation, but you can, or you will be able to see what I'm driving at if I'm talking about generalizable principles that apply to not just parts, but the entire nervous system, maybe. All right, so let's think first about the problem of terminal identity control. And I want to sort of briefly tell you about terminal selectors, but also give you a little bit of a history about it. But again, let's just theorize about the problem of how a differentiated neuron, a fully differentiated mature neuron, acquires its terminal identity features. So if you think about this problem theoretically, and if you think about the differentiated state being defined by many distinct molecular and biochemical features of a neuron, if you think about it like this, you can think about the acquisition of these this differentiated state as being achieved in distinct manners. You can think about each biochemical entity of a neuron to be independently controlled by different sets of transcriptional regulatory mechanisms, right? So this would be sort of the piecemeal model number 2 here. And this is a model that is certainly not unintuitive, simply because if you look at any transcriptome data set of any mature neuron type, you will actually see that any mature neuron type expresses dozens of transcription factors, right? And they should be there for some reason. And one way, again, to think about these many transcription factors and think about regulatory routines is that each one of them has a job in defining unique biochemical or biophysical features of a neuron. But then there's, of course, the other much more simple model, which would posit that those distinct identity features are actually co-regulated by one and the same transcription factor, right? So this is more of a dictatorial model, so to speak. Yes, many transcription factors present in a cell, but maybe some are much more important than others. So this sort of master regulatory model here. So which one of these two models is true? Well, let me give you the historical perspective here, and this is really, I think, a fun perspective. When Sidney Brenner started out with C. elegans, he of course wanted to understand these sort of questions. How does the nervous system develop? How do neurons acquire their identity? What he did in these old days, he and his students and postdocs, was to screen for mutants in which specific behaviors are disrupted. So one very prominent class of mutants that came out of his original screens were these UNC mutants, which where the animals don't locomote properly. Screens done by Marty Chalfie when he was with Sidney identified this gene that is required for mechanosensation, hence MEC. A gene here involved in chemosensation, here one for thermotaxis behavior, here one for ototaxis behavior. What became clear over the years after these genes were identified is that their mutant phenotype pretty much exactly phenocopies the laser ablation, meaning the microsurgical removal of very specific neuron types. For example, Ki1 mutant animals are totally indistinguishable from animals in which you laser ablated that neuron ASE. Same for this, same here, same for pretty much all of these neurons here and genes. And then eventually when those genes were cloned, when the molecular identity was determined, they turned out to be transcription factors that are expressed in those cells. And ensuing molecular analysis by a number of labs, including my own lab, identified that these transcription factors, these classic fate identity determining transcription factors, were actually indeed such master regulatory factors that control the many distinct identity features of a neuron. And I say that not based on fancy schmancy chip analysis, transcriptomic analysis. I say that based on the simple notion that for many of these molecular features, we have GFP reporters available for many years. And when you cross these GFP reporters that measure the differentiated state of individual neuron types into these transcription factor mutants, you find that pretty much any feature you look at is lost in these transcription factor mutants. And a number of years ago, I decided to borrow this term selector genes from the Drosophila literature and just appended this term terminal to it to indicate that what these factors do is they sit at the end of the regulatory hierarchies that generate individual neuron types, in that they initiate these terminal differentiation programs here. Now, importantly, these transcription factors do not usually work alone. They usually work in combination, meaning these are usually multimers, heteromers of several different transcription factors that have this function of working together to turn on these neuron type specific gene batteries. And let's look at this more mechanistically with one example that is particularly well understood. It's a neuron called AIY, an interneuron, a cholinergic interneuron. There are two transcription factors that are expressed in AIY and additional neurons, but these two transcription factors overlap in their expression exclusively in the AIY interneuron. These are very highly conserved transcription factors, by the way. This is a LHX2 or Apteros homolog, a limb homeodomain transcription factor, and this is a paired type homeodomain transcription factor. And again, in each of these mutant backgrounds here, what you will see is that all of these molecular markers that people generated GFP reporters for are lost in either of these two transcription factor mutant backgrounds. And on a more mechanistic level, what we found through promoter bashing, essentially, is that these two transcription factors operate through a cis-regulatory motif that is present in all of those genes here. Neither of these factors alone can bind to this motif in vitro, but together they show this strong cooperative DNA binding to this site here. And this is sort of the mechanistic underpinning of what it means to have a combinatorial code of transcription factors. Those two transcription factors overlap in their expression exclusively here, meaning exclusively here are those genes activated that contain copies of this heteromer binding site in their promoter. Okay, so we've taken this a step further, and over the years, as I said, have aims to not just look at one or two neuron types, but really walk through the entire nervous system to look at such terminal selector type transcription factors. And here is the current status of this analysis. So for the 118 neuron classes there are, we know transcription factors that we think act as terminal selectors for 97 out of the 118 neuron classes, right? And what this table here indicates are these combinatorial codes. You can see here, for example, here's a transcription factor, the distalless homolog or DLX homolog, and an ETS domain transcription factor. And those two transcription factors here operate in this dopaminergic ADE neuron to directly initiate the expression and maintain the expression of these terminal features of these neurons here, such as, for example, the enzymes and transporters for dopamine, as an example. You can see here that there are transcription factors that operate in different combinations. I'll come back to this later on, but I'll give you a preview here of this guy, UNC86, a power homodomain transcription factor, required, as indicated with these boxes here, in a whole different set of neurons to control their identity. And it's pairing up with different cofactors or with different collaborators in different types of neurons to give you the specificity that you need to turn on neuron type specific gene batteries. Okay, so eventually, once this is finished, we will, of course, hope to fill in this table with more factors to get neuron type specific codes of transcription factors for every single neuron class. And in a few minutes, I'll tell you about how we think about these combinatorial codes on a truly nervous system-wide level. All right, so the one thing I want to point out to you, though, is that, as we accumulated that data and we're trying to think about any common themes here beyond the theme of having this master regulatory control mechanism and beyond the theme of having combinatorial control, we were noting that there was one class of transcription factors that clearly predominated the landscape of neuronal identity control. And what I'm boxing here is all of the transcription factors that are encoded by Homeobox genes, meaning homedomain containing transcription factors. So you can see here numerically that they make up clearly the majority of transcription factors involved in terminal identity control. And even in those cases where non-homedomain transcription factors are involved, they usually cooperate with the homedomain transcription factor. Again, this example here of this ETS domain transcription factor working together here with this distalless type homedomain transcription factor. Right. So, and as this became more and more clear that homedomain or Homeobox genes are so important, and I should say that many of these classic mutants that came from the Brenner screen initially, and these are of course totally unbiased screens, right? They were again homedomain transcription factors, and that eventually prompted us to dig a little deeper into these Homeobox genes. I shouldn't really need to give you much of an introduction into Homeobox genes, but I just want to make one thing very clear here to you, which is that, of course, we have all heard about Homeobox genes. Their main claim to fame, not just to fame, but also to Nobel prizes, is this specific subclass of Homeobox genes. It's a derived subclass of Homeobox genes that are clustered in the genome. They're called Hox genes, and they have initially been shown by Ed Lewis and, of course, and many others to be involved in specifying segmental aspects of a body plan. And those transcription factors were, for their initial activity, were identified as being involved in early developmental patterning, right? So these are genes that are transiently acting to organize specific regions of the developing body. And of course, not just in flies, but in other organisms as well. Now, again, as I said, they're only a small fraction. They only constitute a small fraction of all Homeobox genes in any animal genome. For example, here in C. elegans, we have more than 100 Homeobox genes, and only a small fraction of them are these Hox cluster genes. And one question that one can, of course, has, much like these Hox genes have this common theme of acting to establish segmented body plans, is there also maybe, perhaps, a common theme? And what I mean by a common theme is sort of an ancestral theme of what these other Homeobox genes do. And what I want to tell you now about is the work of Molly Rayleigh, a graduate student in the lab, whose work, together with the past functional studies that I just mentioned to you, make a strong case for such an ancestral function of Homeobox genes being indeed in controlling neuronal identity. So what Molly set out to do is to look at this really systematically and very broadly in literally every corner of the nervous system. And what I mean by that is that Molly GFP tagged every single one of these 102 Homeobox genes, and I'm actually noting right now, you probably noticed that as well. I should change this slide. One of these genes here has actually been downgraded recently to be a pseudogene, so it's 102. I should really fix this. All right, so what she did is she GFP tagged those 102 Homeobox genes to look at expression in the nervous system. A little bit of statistics here, about 3/4 of them or so, almost 3/4 of them are phylogenetically very deeply conserved. Others are conserved only in nematodes, and some of them are even C. elegans specific. Now, if you do that, if you tag a gene with GFP, any gene in C. elegans, to establish its expression in the nervous system, that may sound to a non-C. elegans person as a pretty trivial task. At the end of the day, only 118 neuron classes, 302 neurons total. But as any C. elegans biologist and any even C. elegans neurobiologist can attest to, identifying gene expression patterns in the nervous system is far from a trivial task. The way that people traditionally have done that is to hope for the existence of markers that allow to co-stain an expression of an unknown gene with a marker whose expression has been well identified. In the even older days, you needed to do lineaging, look at the history of cells. So it was a very, very laborious task to do this expression pattern analysis. And quite frankly, with no novel tools available, this would have been a task that is essentially impossible. Molly was able to do that because she was lucky to be a contemporary in my lab with F. Yemini, a postdoc in the lab, who developed an amazing tool that we called NeuroPal. I want to tell you a little bit about it. It just came out in last week's issue of Cell. It's incredibly pretty, and I want to tell you a few words about it. So what F essentially did here, he color-coded, he provided a color barcode to every single neuron in the nervous system in a way that every single neuron can be distinguished from one another. That's not to say he generated 302 different colors. That's only to say, and the only is in quotation marks, that he chose colors in a way that every neuron can be distinguished by its color code from any neighboring neuron in the same ganglion. Right? So he built this worm here by avoiding, these are all pseudocolors here, by avoiding the color GFP, which means in practice, you can take this worm where every neuron has its own unique color within a ganglion. You cross it with the GFP reporter, where you see these random green dots here. And now these green dots, the GFP reporter will overlap with a specific color code here, and that will immediately tell you what cell it is that expresses your GFP tag gene. Let's go into the practical details of that, because this is, again, sort of a fun thing that has already shown and will much grow to be a very important tool, I think, in the C. elegans community. So what he's been doing here is he has been taking, or he's making use of 5 different colors. The only color he doesn't use on NeuroPal is actually GFP. But he chose colors that have unique excitation and emission spectra, meaning you can read out colors independently of one another, right? And these are specific fluorophores that he tried that had these specific spectral characteristics. And then the idea is to think about ways how to put colors into individual neuron classes. And here's sort of a simplistic way to look at it. Here's a neuron that expresses only this penneuronal color that we used. Here's a neuron that only expresses this together with a penneuronal. Here's a green one. And here are now overlaps, right? So you can see in this very simplistic way here, you can get these unique color codes by these unique overlaps here of colors, right? And getting a little bit more practical, what that meant here at the end of the day, you take a whole bunch of promoters from different genes, each of which expressed in subsets of neurons. And if you put them all onto the same transgenic array, you'll get these in sum, you'll get this color coding here. And this is how it looks in practice. You have 41 different reporters listed here. Some of them strongly, some of them weakly expressed, which again is information in terms of giving you a specific color combination with other colors together. And that gives you this beautiful color code here at the end of the day in a single worm, right? So this is a single transgenic worm that Molly then used to cross her GFP reporters into, and then she could identify uniquely where all of these Homeobox genes are expressed in. I don't really like to diss other things, but I do want to point out that some of you may have a word swirling around in your head right now, which is rainbow. It's a very beautiful transgenic mice that Jeff Lichtman generated many years ago. The key difference here to rainbow is that rainbow is a stochastic labeling, right? So it's not deterministic. Every animal looks different. And you really can't tell which cell is which because, again, the labeling is stochastic. This is not stochastic. This is highly deterministic and highly reproducible from animal to animal. The color code is totally stable. All right, so this is sort of a little bit of background to make the non-C. elegans people become a little jealous of what we can do now in C. elegans. Okay, so here's what Molly found. She found that using NeuroPal to identify the expression pattern of all these 102 Homeobox genes, she found that 80% of this gene family is expressed in the nervous system. So this is already very striking. But then she also found that while there are some that are penneuronally expressed, and a subset of them actually exclusively penneuronal, meaning only in all cells of the nervous system but nowhere else, while those Homeobox genes are actually ubiquitously expressed, she found that the majority of them are actually quite sparsely expressed. Anything between a few to 10, 15 or so neuron classes. And if you look at this cell by cell, so this is listing every single neuron here. This is listing the number of neurons, the number of Homeobox genes. What she found is that every neuron expresses this range of Homeobox genes from 1 to 10 max, usually around, on average here, 4 to 5. But the really amazing thing of this analysis was that she found that every single neuron class in C. elegans expresses a unique combination of Homeobox genes. Meaning every single neuron class can be defined not necessarily, well, you can of course define it in many ways by its gene battery, by hundreds and thousands of genes, right? But you can essentially boil this down to a definition of neuronal identity by the combination of Homeobox genes that it expresses, right? I should say, you may have remembered that I told you that there are more conserved Homeobox genes, that some are more conserved than others. And these neuron type specific codes, you actually get them by the conserved Homeobox genes alone, right? All right, so what about functional analysis? Well, I did sort of the unusual way. I told you about functional analysis first, right? I told you about all of these terminal selectors that came out of genetic screens. These were functionally defined. I told you many of them were Homeobox genes. So we had the functional analysis first, so to speak. And now with this expression analysis, we broadened this to essentially the entire nervous system. And of course, we're doing more mutant analysis now. And we're hoping, actually now we're really expecting to find that when we do these Homeobox gene mutants for every single neuron class, we will indeed find that every single neuron class in C. elegans requires a combination of Homeobox genes to acquire their unique identity. Okay, so what I want to now walk to is or what I want to now come to is again, a mutant analysis of a Homeobox gene, but now looking at this problem from a somewhat different angle that at first may not be intuitive, but if you think about it, and if you hear me ask the question, it's actually a very obvious and intriguing question. And this is work by Emily Burkhoff, a graduate student in the lab. And what I'll tell you about is this Homeobox gene, conserved Homeobox gene called UNC42, which is expressed in a whole bunch of different neuron classes, right? And again, in each neuron class, it's part of a neuron class specific code of different Homeobox genes. So here are all the genes that, all the cells that express UNC42. It's a total of 15 different neuron classes. And the question that I want to discuss with you, first of all, is again, the functional question. What is the function of UNC42 in these 15 neuron classes? Is there a common theme in the function of UNC42 in the different neuron classes? And in fact, is there a common theme that unifies somehow these 15 neuron classes? I mean, why is it that these 15 neuron classes express UNC42, right? Why isn't is UNC42 totally randomly used by a set of neurons? It's just sort of a evolutionary happenstance that you include UNC42 into your combinatorial code? Or is there something about these 15 neuron classes that predestines them to utilize UNC42 for whatever they do? So let's look at these 15 different neuron classes. So these 15 different neuron classes that express UNC42 are not at all related by lineage. Right? I'm showing you that in this sort of very condensed theme way here, where I'm indicating every single neuron class in C. elegans, well, except the very small handful that come from the P lineage here, as a color bar. The color itself just gives you a random identity feature here, the neurotransmitter identity. So these are all the neurons in the C. elegans nervous system. You can see that very different types, cholinergic, glutamatergic, peptidergic neurons express UNC42, and they have no lineage relationship to one another. So it's not simply the case that these 15 neuron classes that express UNC42 are all linearly related, and because of that linear relationship, turn on UNC42 expression. That's not the case. However, if you look at these UNC42 neurons in this manner, something very striking pops out here. What you can see here is that all of the 15 different neuron classes that express UNC42 are synaptically connected to one another. Again, these are very distinct neurons. There's a glutamatergic sensory neuron here. It's actually the only sensory neuron here. There's a bunch of interneurons here. Interneurons are the hexagons here. And you see all the so-called command interneurons here. And you see a bunch of motor neurons, actually mostly head motor neurons that innervate head musculature, right? Many of these neurons had actually previously been implicated functionally in a nociceptive circuit. This is actually a really interesting polymodal nociceptive neuron in C. elegans that responds to a variety of different chemical and also mechanical stimuli and induces a complex series of reversal events, right? And these neurons are functionally involved in that. So again, this is a distinct set of neurons, but they're all unified by their mutual synaptic connectivity. And that, of course, now raises the question, is UNC42 only specifying the identity of these individual neurons in terms of their molecular gene batteries, and or is it also controlling the ability of these neurons to function as a circuit and maybe to assemble as functional circuitry? And let me walk you through this analysis. So I'm going to show you in the next couple of slides, again, this circuit here. I'm going to show you how things look in wild type and how they look in UNC42 mutant animals. And what I'm color coding here are different molecular identity features that we're looking at. So we're looking here at the expression of a gene called EAT4. It's a vesicular glutamate transporter, vGlut, that is expressed in these two neuron classes of that circuit. And this is the vesicular transporter for acetylcholine, which is expressed in those neuron classes here. So we're basically now asking, is UNC42 required for these cells to acquire their unique neurotransmitter identity, and hence the ability to communicate with one another? And what you can see, indeed, in UNC42 mutant animals, you see a loss of neurotransmitter expression here in all of these neurons here. I don't think I'm showing you the data today for this, but we can also eliminate AVA expression if we combine UNC42 with another transcription factor, and I'll actually mention that later on. Okay, so this already tells us, all right, so if you don't have UNC42, you're pretty screwed up in terms of being able to communicate with one another. Now we're going to look at neurotransmitter receptors. And again, now we see a complex expression of these different types of glutamate and acetylcholine receptors. These are all ionotropic receptors. Well, most of them, not all. And you can see that, for example, this neuron here expresses, 5 different neurotransmitter receptors here. We look at the mutant, and again, the majority of these genes are lost in UNC42 mutant animals, right? So these neurons are not just defective in sending signals, but they're also presumably quite defective in receiving signals and processing information on this receiving end here. Let's look at neuropeptides and neuropeptide receptors. Same color code here, bunch of neuropeptides, bunch of receptors here, very specifically expressed within that circuit. And you can again see very striking effects of loss of UNC42 on these signaling markers here. Okay, so we're basically where we were already in the beginning of my talk. Yes, we think UNC42 is a terminal selector. It controls these many different identity features of a neuron. I just showed you the signaling part of it, but there are other features. I can take you through this in a few slides. But I want to already preface that by saying, by quoting here from this review a couple of years ago. Let me just read this out here. And I think, although this is 8 years old, this is still sort of a statement that would apply these days even more so. So what these authors said here is that there appears to be a common strategy to generate neuronal diversity across the central nervous system, when the combinatorial expression of different transcription factors specify regional and subtype-specific neuronal identity. So that's what I told you in the first part of my talk. These are essentially terminal selectors here, right? But the question ever since has been, however, an open question is whether this process also encodes the molecular identity responsible for connectivity. Right? So this is exactly sort of the more broad nervous system-wide view, where you don't just look at neurons in isolation, but you look at them in the context of circuitry. So what does UNC42 do in terms of controlling the assembly of these neurons into functional circuitry? So I'm going to show you some gruesome work that Emily did. It took her almost 2 years. It's not pretty. But it's the ultimate thing to do, which is EM reconstruction of UNC42 mutant animals to look at synaptic connectivity, right? To ask, is UNC42 also required to assemble neurons into circuitry? Now, of course, you're asking yourself, well, why the heck did we do EM for this, right? Why don't you just mark synapses in some other ways? Well, here's the problem. The problem is that if you mark synapses with reporter gene technologies, such as, for example, GRASP, GFP reconstitution across synapses, well, you need drivers to express things in specific neurons. But I told you that UNC42 controls the expression of many of these cell type specific drivers. So it's very difficult to fluorescently label neurons in UNC42 mutant animals, right? And EM, of course, doesn't do that. EM is totally blind to any fluorescent reporters, right? You basically just look at anatomy, right? So what Emily did, again, very courageous and facilitated by the fact that that Sidney Brenner's lab decades ago did actually already fix UNC42 mutants. So Emily really only, in quotation mark, had to section them. It's done in collaboration with Dave Hall's lab. And then trace and establish connectivity. And what Emily found is shown here. It's again not intuitive from looking at this. Let me just explain to you what you see here. You see the tracing of the processes three-dimensionally. And the point of this slide is that the overall neighborhood of the processes is preserved in UNC42 mutant animals, right? So these neurons that don't have UNC42 still send out axons, and they're still neighbored by the same type of neurons that they're neighbored by in wild-type animals, right? This is, again, showing all of them, and this is showing a specific subset here. Those two neurons, they normally neighbor one another, they overlap here, and they still do that in UNC42 mutant animals, right? And this is, of course, now the prerequisite to ask, well, what about synaptic connectivity, right? And here's the nice thing. What Emily found is that if she looked at synaptic connectivity of neurons that express UNC42, the gray shading means pre and post-synaptic expresses UNC42, you will see a reduction of synapses between these neurons in UNC42 mutant animals. Okay, so let me be totally honest with you. There's a huge problem here. And the huge problem is this is an N of 1. You can easily get me to talk for about half an hour about connectomics. I have strong opinions about that. Let me boil this down into a 30 seconds diatribe. EM analysis will always have this problem of limited sample size. It's a huge problem. It's particularly a problem if you think about sinking millions, if not billions of dollars into doing connectomes of mouse or even human brains. Everyone is keen on making the first connectome, but as we have well seen in C. elegans, nobody is keen on doing a second or third or fourth connectome, right? In part because it's so laborious, right? So, and this is of course the problem with this data. This is really just a single animal. And for all of the science neophytes, the students, I don't need to tell you that, but I'll say it nevertheless, an N of 1 in biology is never really good. You want to strive for more than that. And we need to strive for more than that. We still haven't figured out, we certainly don't want to do more EM, so we have to simply think harder about how to label synaptic contacts in wild-type and mutant animals. And there is actually one type of synaptic contacts that we can relatively easily look at, and Emily has done that, and these are electrical synapses, right? So this circuitry here is not only composed of chemical synapses, but also plenty of electrical synapses, right? Gap junctions that are made between these neurons in that circuit. Now, those synapses are actually quite easy to look at because gap junctions or electrical synapses are homogeneously made by just one type of proteins. Oops, wrong slide here. By one side of proteins, which is annexin proteins. They're called connexins in vertebrates. And Emily looked so far at 3 of them. You see they have specific expression profiles here. And again, what she sees is an effect of UNC42 on the expression of these electrical synapse proteins, right? So that, of course, leads to the very clear prediction here that indeed electrical synaptic connectivity should be pretty messed up or is pretty messed up in UNC42 mutant animals. In terms of chemical synapses, what we are sort of stuck with at this point is to look at candidate synaptic recognition molecules. And again, Emily has started to do that. She looked at a whole bunch of immunoglobulin superfamily members, also other proteins such as netrin, a protein, of course, also very important in synapse formation, not just axon pathfinding. Again, there's nice combinatorial expression of this family of these Ig chems here in UNC42 mutants, and again, very strong effects of UNC42 on the expression of these putative synaptic recognition molecules. So, so this is basically what I want to tell you in terms of primary data here. I've told you that UNC42 affects many molecular features of these neurons, and I've told you this preliminary data that suggests that connectivity in this circuit is also very much disrupted in UNC42 mutant animals, in part by this quite insufficient EM analysis, but also by the analysis of the molecules that we think are involved in establishing that circuitry here. So this is fine and good, and this, of course, very nicely insinuates that the logic behind using the same type of transcription factor in different neuron types to control identity, it suggests that this logic of using the same type of transcription factor has a role in assembling neurons in a circuitry. But again, there's some sort of a problem here because these neurons are different from one another, right? ASH is, both of these neurons require UNC42, but they're different neurons, right? Now, simply remember to what I told you in the beginning. Well, that's presumably because UNC42 pairs up with different partners, homodomain transcription factors and others, to make ASH and AIB become different from one another. And that's exactly what we see. For example, we know that this transcription factor here, UNC3, is working together with UNC42 to specify the identity of those 4 neuron classes, right? So we think that those 2 factors combinatorially act to activate the gene battery here that unifies these neurons, makes them command-into neurons, right? There's another transcript, and by the way, there is actually some data here that actually very nicely shows this collaboration of these 2 transcription factors. You see that either mutant alone here has somewhat lame effects on the expression of, I'm sorry that this is cut off here, this is expression of the UNC17 vesicular transporter for acetylcholine. There are effects, but they are just a dimming of expression, partially penetrant. You build a double, you now see an almost complete loss of this cholinergic identity in these mutant animals. There's another transcription factor, CFI1, that is required in this neuron class, and presumably defining the distinct identity of AVD versus AVA in AVB. We've identified another, sorry, this is actually not us, I'm sorry. This is Bruce Whiteman's lab, who had shown before that AVK is specified by FEX1, which again, we think works together with UNC42. Henrik Brinkman's lab has shown that this Homeobox gene is involved in specifying this identity. And again, we think it works together with UNC42. And we found recently that these 2 transcription factors, again, Homeobox genes, work together with UNC42. Right? All right, so here's where things become even more interesting. Each one of these transcription factors itself is expressed in additional neuron classes. For example, UNC3 here is not only expressed in the command-into neurons, it's also expressed in ventral nerve cord motor neurons. And these are, again, synaptically connected with one another. So maybe we can think about circuitry as being assembled by transcription factors that work in synaptically connected neurons. And each one of these transcription factors works in partially overlapping sets of synaptically connected neurons to assemble neurons in a circuitry, but then give unique identities to individual neurons within a circuitry. Let me schematize this in the following way. So this again would be a transcription factor, UNC42, specifying the identity of these 4 synaptically connected neurons. These are synapses, these are different neurons, right? Here's another transcription factor that now is co-expressed with UNC42 in this guy. And hence this neuron is made different than this and this. But this transcription factor is also expressed in those guys here. And those neurons are again made different by this transcription factor working with another one that is again expressed in an overlapping set of neurons here, and so forth and so on. So you can again think about complex wiring diagrams as being specified by these individual circuits here, by individual transcription factors that join together here to build this overlapping circuitry. And this is how it looks for those guys that I just mentioned to you. In the center here is this ANC42 circuitry, right? And then there are all of these combinations here. This would be a combination of 3 different transcription factors, ANC42, ANC3. ANC3 is expressed on all of these blue neurons that generates additional circuitry, right? So meaning those terminal selectors that are expressed in synaptically connected neurons, we call them actually now circuit transcription factors, because they don't just identify or specify unique molecular features of a neuron, but also specify the ability of these neurons to be assembled into functional circuitry. And I'm just realizing right now, and I'm really annoyed by that I missed a slide. And this would be my last slide, and I'm just now going to verbalize this. One way to think about this here is to think about this in an evolutionary context. Maybe with the employment or the frequent re-employment of Homeobox genes in specifying neuronal identity and connectivity tells us is that it was maybe an ancestral theme in a very simple nervous system to employ a Homeobox gene to specify simple synaptic signaling features, and maybe to assemble via the expression or the control of expression of the homophilic adhesion molecules a set of very similar neurons into sort of a nerve set of a very homogeneous group of neurons. Maybe this was sort of the Ur-Homeobox gene, the very first Homeobox gene that was employed in that function may have specified signaling capacity and assembly of cells into a simple nerve net, right? Think about this as sort of the most ancestral state. And then think about gene duplication and diversification, right? You get more Homeobox genes duplicate, the expression pattern diverges. They may now recruit different types of adhesion molecules as their target genes, and thereby diversifying synaptic connectivity, right? So I'm basically following here the Dobzhansky's view of nothing makes sense except in the light of nothing makes sense in biology except in the light of evolution. Maybe that's how to see Homeobox genes as an indication of how very simple nervous system were patterned. And what we see here with these circuits here is sort of these remnants of these ancestral Ur circuits, so to speak, that have become more and more diversified and complicated via gene duplication and divergence. So that's one way to think about this, and of course there are very interesting questions in terms of how to further provide evidence for such an evolutionary model, and I'm happy to discuss that maybe in the Q&A. So let me just thank the people who did the work. I'm actually not listing here many past members of my lab who contributed individual data points to neuronal identity control by these terminal selectors. And I'm doing that because I really want to highlight those people here that have broadened this analysis to a true nervous system-wide analysis. And that's two former postdocs in the lab, Laura Pereira and Esther Serrano-Saiz, who were really the first to take a systems-wide approach by mapping neurotransmitter identity throughout the entire nervous system, which provided us with a great tool to look more broadly at our transcription factors for how they control neuronal identity. Then, of course, Molly Riley for broadening this Homeobox gene analysis. And her analysis would have been barely possible without F. Yemeni's NeuroPal tool. And Emily Burkhoff, as I mentioned to you, was the driver for this project on circuit transcription factors. All right, so let me actually get out of my shared screen. I hope I'm doing that right so that I can see people more. Click to close message, close video. And now I'm very happy to take any questions.
So that was unsurprisingly wonderful, Oliver. Really nice.
Thank you, Bassem.
Really clear and didactic. I truly enjoyed it. So we've got a couple of questions, and I have a question of my own, but I'll start with the questions from the audience, so to speak. There's one question in the chat and one question in the questions and answers. Okay, so now we're getting more questions. Okay. So the first question is from Haluk Lajin. He is asking, how often do you get UNC42-like severe phenotypes in single mutants of other factors? So in other words, how general is the severity of the UNC42 phenotypes? And in a way related to that is, so the question continues, how much redundancy, therefore, is there in the system?
Yeah, these are great questions. So it really depends. So in some cases, in some neurons, this is actually a complicated question because it gets really into transcription factorology, into the biochemistry of how transcription factors work. So if a transcription factor works in a truly cooperative manner, right? In those cases, you remove one protein from the code and nothing works anymore, right? Because there's no more DNA binding of any other accessory factor, right? In those cases, there's no redundancy whatsoever, right? The neuron totally breaks down functionally. And we see that in a whole bunch of these UNC42 positive neurons. ASH, for example, you kill it, you look at UNC42 mutant animals, it's identical, right? It's totally penetrant, right? But then the other way how transcription factors work is a more distributed way. It's a so-called billboard model of transcriptional control, where you have transcription factors binding independently to DNA, independent of one another, right? So meaning you take out one, and depending on how many binding sites you have for the other guy, you can still get some extent of expression of the target. In some cases, actually, really a lot, meaning you may not see any of an effect, and you really need to remove both before you see something. And depending on the neuron class, we see a mixture of things. I would say still that the great majority of cases, it's really more in support of this cooperative model, meaning you don't see much redundancy, but there are some cases where we do see clear redundancy, which again we think is because of that different means how transcription factors operate on DNA. Bassem, I'm not hearing you.
Oh, sorry, I forgot to turn on my mic. Yeah, sorry. So Fikri Selimi is asking, one would predict that loss of one of these Homeobox genes would transform the code. And so at least some of the examples, and I think I agree with the logic there, ought to be gain of features, and not just always loss of features, right?
Yeah, so, a great question.
Of that.
Fantastic questions. We do see it, but extremely rarely so. And we can actually predict that based on the code, because if you compare the codes, there are very few examples where the code just is different by a single gene. Right? That's not to say that we don't see homeotic identity transformation. In some cases, we see them. And actually, in some cases, we have characterized them really in depth, the extent of homeotic transformation. And we see that sometimes. Right? But in the vast majority of cases, we get these, very undefined states of these neurons just don't differentiate. And I want to add here one thing, because I'm sure there are lots of non-C. elegans people here. In vertebrates, this is a phenotype that's very hard to capture. Yeah. Because if you are a neuron in vertebrates that doesn't talk to your partners, you usually then don't get any trophic support and you'll die. Mm-hmm. In fact, we went to mouse to look specifically at the function of one of the homologs of our genes. We published that 2 years ago, a gene called BRAIN3, the UNC86 homolog. And as the elegance geneticists, what we had to do to analyze these mouse mutants was we had to build a double of BRAIN3 mutants with a mutant in which cell death was suppressed. And only then did these non-specified neurons survive. And we could indeed say that, like in C. elegans, this transcription factor is required for driving identity, and you don't automatically switch to another identity. Yeah. You can, but it's pretty rare.
Yeah, that's great. Yeah. So Daz is asking, you mentioned pairs are common motifs in neurons. Do you get triplets? I'm not sure. I guess this is referring to the numbers of a certain neuron.
Yeah. Yeah. So we have many cases where there are more than two. In fact, the majority of cases are more than two. And in one case, we actually have looked at this past work from Nuria Flames, a former postdoc in the lab, to really dissect the cis-regulatory architecture of these genes that have multiple transcription, more than two, right? And we can again see what we said before. You need to knock out on the promoter level, right? Many of these very different binding sites before we, or several of them, before we see an effect. So combinatorial codes does not mean just two. It often means much more than that.
So I think this is from Sophie Jarriot, who's asking, hi Oliver, nice talk. Could you tell us if you went the opposite way and selected synaptic circuits first and then looked at their TFs? So rather than asking, do neurons that express the same TF connect?
Right.
Are all connected neurons expressed? I guess. Do you always find one/a few homeobox genes in all these neurons?
Yeah. So no, that's a great question. So and hi, Sophie. So here's how we did it. I mean, as you know, there's really no such thing as very neatly defined separable circuitries. It's just all a big hairball, right? And if anything, this UNC42 one is the one that comes closest to something that you could call sort of an autonomous circuit. But here's how we did this, and I think this is actually the most definitive way to do that. We simply use statistics. We simply ask for any homeobox transcription factor, or for any transcription factor where we know the expression pattern of, and that is expressed in multiple neurons. We simply ask statistically, is the extent of connectivity between these five neurons that express that transcription factor, higher than you would expect by chance, right? So, we needed to talk to statisticians about this. It's not totally trivial, and I'm not sure I can explain the math behind this to you, but I can tell you that you can get a statistical analysis of that, and that led us to identify, we have a number of about 20 or so transcription factors, most of them homeodomain transcription factors, but some of them not, like UNC3, that actually do display that they are the connectivity of the transcription factor expressing neurons among themselves is higher as expected by chance, right? So, but that brings me to an important point, and I want to make this very clear. I do not think, and I don't want to be remembered as saying that every single connection in the C. elegans connectome can be explained by that logic, right? I would argue that what we're basically just seeing here, we're barely seeing, right, is sort of some core circuits, right, that you can assemble with that circuitry. And that's how I sort of view that. I view that in an evolutionary context, right, that this is sort of a window into the past. This is what, UNC42. And those UNC42 neurons, or all of these connected neurons, interconnected neurons, they may have been ancestrally very similar to one another and only diverged over time. So this is sort of the idea. And I know it's a lot of loose talk here. And just to you, Sophie, I don't know whether we talked about this before, but the way we're addressing this sort of evolutionary aspect is by looking at a very distant nematode species, Pristioncus, where we are almost finished in doing the connectome. We're going to look at transcription factor expression, including your friend UNC3 also, and look whether this statistics still holds, or whether we can really see co-evolution of new synaptic connections or loss, and this correlates with the gain and loss of these transcription factors.
No, I think I really like that, and I don't think it's loose talk, because it's exactly what you said at the end. I mean, obviously we all know it, but we tend to forget it when we do experiments. The nervous system of any given animal is built on the nervous system of its ancestors. It's not invented and new every time the thing develops. And so, if you go back to a place where there were fewer neurons and fewer sort of genes, or perhaps less expression, then it becomes almost automatic that the first set of connected neurons would have shared as sort of a code, a transcriptional code. But I have a question about that that's sort of tangential to it or orthogonal to it, maybe. So you mentioned something very interesting, which is that these neurons are not related by lineage; they're related by circuitry. And what I missed in the talk, and maybe I missed it, and it was there, is sort of the temporal aspect of when do these transcription factors begin to express in these neurons compared to when these neurons wire up or express certain features or whatever. Is this really a sort of a, linear, this first, this, or is there a lot of, feedback where you sort of burst a certain factor that favors a certain feature and that feature reinforces the factor and so on and so forth?
It's a great question, and you're putting your finger into a very deep wound of C. elegans biology. And what I mean by that is, there's really a black box problem in C. elegans. So let me give you the temporals here. So most of the neurons in the nervous system, the embryonic nervous system, are born at around 400 minutes of embryonic development, roughly, right? The animal hatches at 800 minutes of development. And that's basically when people then look and see in an L1 larvae, here are all the synaptic connections, right? But in this window here of when a neuron is born, and we know that this is when terminal selectors are turned on, at about the time the neurons are born, maybe a division earlier, right? But then this window of, is there exactly this sort of fine-grained temporal succession of how circuitry is built is totally unknown. Right. So at this embryonic stages, worms are very hard to look at. They very vigorously move in the embryo. So there's very little known about the emergence of these connectivity patterns. And also because of the way we do stuff with GFP reporter gene technology. And we're talking about very fast time windows here, right? So it's very difficult. But you're absolutely right. This would be also my bet as being a very important part of the logic. Who comes first, who grows out first, who wins out in expressing one transcription factor earlier than another. But this is actually really very hard to tackle in the worm.
But I think we'd agree that it's really, really critical, because I think nowadays we use terms like stochastic or deterministic or whatever without really, and my hunch is that we're essentially all referring to these sort of feedback probabilistic processes that, that at the end of the day are the same logic across all examples. We just don't have the temporal resolution and sort of to look at them. Okay, so I guess related to this in a relatively important way is a question from Jihao Liu, who says, thanks for the beautiful talk. Is it possible that the identities of the neurons are determined by their birth order during development, and that the neurons that are born together, so perhaps by spatiotemporal proximity, therefore, are wired together, and that's why you can see that the neurons wired together express the same transcription?
Right. Again, a good question. It can't be as simple as just born together, wired together, right? And I know this is sort of a view that in the Drosophila field, has taken some hold in some lineages, which is good. I don't doubt it. It's just, the time window is so narrow in which some neurons are born, and if you, actually, here's the most trivial example. If you just look at sister neurons, right, that are born at obviously the same time, right? They're usually, they're not, they have no preference to be synaptically connected to one another compared to something that's born later. So again, I don't deny the potential importance of temporals, but we just know really too little about it.
Okay, so a question by Frank. It doesn't say Franco. I don't know if it's Frank Pouleux or another Frank, but anyway. Fabulous talk, Oliver, as usual. How do you think about the evolutionary constraints of these transcriptional mechanisms, thinking about it from the evolution of the TF binding site, which ultimately determines the downstream targets?
Yeah. So, I think that this is I would give this as an explanation as to why you have such a master regulatory model, that you don't have actually, that this is a highly evolvable system, right? By having, a relatively simple combination, you can envision that it is pretty easy to gain or lose responsiveness to a set of transcription factors in a cell, right? And that this basically drives novelty and drives selectability, so to speak, right? Again, this has actually not been really well addressed in practice. And this is something by going to actually a much more closely related nematode species that a new postdoc is doing, looking at C. brixae and very specifically looking at targets of individual terminal selectors and looking at divergences and see whether this is really true, that you have this sort of breathing, that you gain and lose targets rapidly. There's one problem with that argument, though, and it's a very interesting problem, which is these sites, even for combinations, they have pretty low information content, right? So you can find them actually a lot in the genome. So there has to be some superimposed layer of control, which could be relating to earlier events and chromatin accessibility that, you need to sort of generate some sort of playing ground in which, meaning accessible enhancer regions in which you can then sort of evolve motifs. But there's probably more than just that.
Well, so along those lines, then, would it make sense to suggest that, in fact, the evolution of complex combinations and partially redundant combinations, given how degenerate the binding site code is for the vast majority of transcription factors, actually ensures that you have a playground, but it's not that you have novelty, every 2 seconds, because.
That's right.
Because unless all the factors are changing simultaneously, you really won't get novelty. So in a way, you create a massive field of novelty to happen, but in fact that novelty isn't at the genome sequence level, but that novelty isn't acted upon because of the constraints that you need all the redundancies to work out at the same time. Does that make sense?
That's very well put. And let me extend this thought to a specific class of genes that we looked at, which are pan-neuronally expressed genes, right? So we're talking now about synaptic vesicle machinery, things that neurons absolutely need. So we actually studied them, and we still do, very extensively. And what we find there on the cis-regulatory level is something very striking. There we find extreme and excessive redundancy. Meaning we have not yet identified a single transcription factor that gives us any mutant phenotype in terms of regulation, in terms of direct activation of pan-neuronal genes. If you go on the cis-regulatory level, we see this redundancy played out by incredibly redundant promoter sequences, where totally different chunks of the promoter, with no sequence similarity, give you these pan-neuronal patterns. So here we have a case. You can't fuck around. Sorry, you can't screw around with pan-neuronal genes. This is not something you want to evolve, right? So, and this is exactly what you said, right? I mean, they are basically the constraint. This system is totally constrained, right? You don't evolve that, right? And yes, so is this other, neuron type specific logic is exactly sort of this good balance of, having some robustness, but not too much.
Yeah. No, it's funny because Oliver and I, before coming online, we're talking about, online versus meeting, in conferences, and where the action in conferences happens because you've got 5-6 people around a bottle of wine, and the talk starts flowing. And I guess we got into that mode now.
That's right.
Anyway. So Oshri Abraham is asking, do you think that those identity TFs have also a role during plasticity, neuronal plasticity?
Yeah. So so this is actually what about half my lab studies now is how gene expression, how these hardwired gene expression programs get modulated based on the following parameters. One is time, right? How do these differentiated state of a neuron change in larval and adult development by sex? How do, when an animal sexually matures, how do individual genes within very specific neuron types change their expression, or under environmental conditions, right? And there's a bunch of striking examples that we have, and the punchline is actually quite simple. for everything we have seen so far, which is for any plasticity gene, meaning any gene that is turned on under specific circumstances, right? Be that external, be that on the time scale, be that on the scale of sex, they all require terminal selectors. But obviously, terminal selectors are not sufficient, right? Because this feature is not a hardwired feature. It comes on only in specific conditions. So these are all promoters. The promoters of these genes are tuned such that they require terminal selectors, but they're not sufficient. What they require to be turned on is an additional transcription factor that then works with the terminal selector to turn on a gene. One specific, the most beautiful example here is hypoxia. Hypoxia has some very specific impact on some very specific neuron classes. There's one chemosensory neuron that becomes serotonergic under hypoxic conditions. We find that this requires HIF1, hypoxia-inducible factor, but it also requires the resident terminal selector in that neuron, right? So that resident terminal selector is not sufficient to do it. It requires HIF1 as a cofactor to become responsive to a specific cue. So plasticity, yeah.
No, so that's almost like the notion of pioneering, not in the chromatin sense necessarily, but at least in the larger sense of it, that we often talk about in early neurogenesis. So you need the terminal selectors to pioneer accessibility of environment-dependent genes.
That's exactly right. That's exactly right.
That makes sense. That makes a lot of sense. And actually would make a lot of sense for how you can genetically encode development that is nonetheless has to deal with an unpredictable environment in the future.
Right.
So.
That's right.
Okay, so the last question we have in the box is from Suzanne Faulkner, and it's, how do you think about the hen and egg problem in circuitry? For instance, does UNC42 instruct neurons in a wider sense to form a functional / behaviorally relevant circuit? Or is the common expression the effect of multiple neurons first connecting and then working together? And it's kind of back to the issue we were discussing earlier.
Yeah. Quick question. I have a pretty definitive answer for that, which is it's not the latter. Connectivity does not instruct for example, the induction of UNC42 expression. And I say that for actually, this is actually pretty easy to test. If you take a mutant that is totally fucked up in connectivity, you take something like UNC6, sorry, I'm again falling into the beer mode, which I know is culturally a little problematic, but anyway. If you very severely disrupt connectivity by taking an axon pathfinding mutant, right, and things are totally messed up, right, and then you look at the expression of not just terminal selectors but also many identity features of a neuron, totally unaffected, right. So connect, at least in C. elegans, right, the connectivity really is secondary to identity acquisition.
And I think that's fair to suggest that in most models that people at least study tends to be the case, not just in C. elegans. But okay. Well, we got just another question. I don't know how you're doing on time, Oliver.
I'm good.
Okay.
Yep.
So Nadine Randall is asking, in the transcription factor mutants, do you also see effects on the pathfinding, i.e. on neuronal development? Right?
Can you actually repeat this? I had, again, this funny noise coming up exactly when you talked. So can you say it again, please?
Yeah. So the question is whether in the transcription factor mutants you also see effects on pathfinding.
Yeah, I couldn't hear actually a word. Is there any way I can see this?
Hang on. Yeah.
Here. Ask a question 10. Wait, unanswered question. Oh, wait. Good. So wait.
Here.
Is it the last question by Nadine Randell? Okay, great. And you can hear me fine. Good. So the question is, in the transcription factor mutants, do you also see effects on pathfinding neuronal development? Yeah, a great question. And not really. They look a little messed up, but they still do pretty much what they're supposed to do, right? I mean, if you think about, let's say a sensory neuron with a dendrite and an axon, you look in a terminal selective mutant, it pretty much still looks like this, but they're probably very subtle defects. I mean, as I said, in terms of connectivity, but there's no gross effect on pathfinding, which, by the way, is very consistent with the fact that exon pathfinding genes are usually transiently expressed during development, right? And this is not terminal selector stuff. Terminal selector stuff locks things into place, right? So you almost don't want an exon pathfinding gene to be under control of something that is so, rigidly hardwired, if you want. So that's basically how we think about that.
Yeah.
Having said this, what I just said is based on a relatively limited sample size, because in many terminal selectors, we can't look at the mutant because all of the markers are off, right? So it's very hard to visualize them. So I'm inferring that, from looking at, in part hypomorphic alleles, and sometimes we can actually see still a little bit, maybe one or two markers left, and that allows us to look at the neuron. But by and large, we think it's looking good.
Okay. Well, that concludes the Q&A. Can you hear me?
Yes, I can hear you now again.
Okay.
Yep.
So that concludes the Q&A. Thank you very, very much, Oliver, for just a thought-provoking and a lovely talk, and I think wonderful discussion. Thank you all for attending. Thank you to those who asked your bright questions. They were very cool. And see you again soon on this very forum. Oliver, looking forward to having a beer and not restraining yourself from saying what you really think.
All right. Thank you. Good to see you, Bassem. Thanks, everyone.
Bye-bye.
Okay.